take the nectar off of, oh, sorry, um, that this sugar will attract these other insects and they can act as kind of like a natural biodeterminant, like deterrent of like other species, especially the hornets in terms of like not being either parasitoided or being predated on by other species. So for the three species I'm trying to delineate here, we have Disholcaspis malefica, Disholcaspis prehensa, and Disholcaspis eldoradensis. It's important to note that all of these share a very similar morphotype, with prehensa kind of being the biggest standout. And all of these galls kind of show plasticity in like how they are displayed. Um, so basically, the question I was trying to answer in my thesis was, are these species actually distinct? Because they all seem to form on very similar oak trees and also have very similar morphologies, or are they all the same species, or how are they best described to actually delineate them? So, as some background, the first people to start to like describe these species back in like 30s and 50s were mostly Stanford scientists kind of walking around California and noticing these growths on oaks. And so we had these three descriptions kind of written down and then added to kind of like the taxonomic groups back then, and these were all identified interestingly by the actual galls and not the actual insects themselves. So there was a lot of pushback of like uh, that these galls, which all kind of seemingly look the same, are different species that are on different oaks, but traditionally through the use of molecular methods with other similar cynipids in Europe, they've been delineated against trees. So it's interesting that in America, they seem to be on many different oak trees when mostly these wasps like to specialize. And so my question is trying to like figure out if these are even different species to begin with, and if they are they host tree specific or are they just kind of looking different? Um, so first, when we were going to collect these species, we looked at iNaturalist, and we're seeing that however iNaturalist is keying out these species, it seems Malefica has this large range. Eldoradensis seems to be pretty localized in California, and then Prehensa has the smallest range, kind of around the California Central Valley. And then to look if to see if their actual gall sizes were different or if they were controlled by different things other than their genotype, we looked at average gall sizes taken at different collection plots, and then we put that over the average minimum temperature of the closest NOAA node to the collection site. And we were able to see that with our galls that we collected, that there seems to be a pretty good relationship between temperature control and the size. And so it's not part of the genotype expressing itself that like Eldoradensis has bigger galls, and it's usually just all of them will make bigger galls if the temperature is higher. To further kind of confirm this, we used a lot of iNaturalist observations and went in and measured using ImageJ to do proportions of the gall height to the gall width. And using that and using more NOAA data, we were able to get a larger data set rather than just our collected galls using iNaturalist. And this also confirmed that there seems to be the relationship between size seems to be with temperature and not controlled purely just by the genotype. To further see if these species were actually genetically different, we first took our wasps, we extracted their DNA, then ran initially two DNA barcodes. We sent those off to Eurofins and then used PhyML at the South of France bioinformatics platform, which we were able to make gene trees. And we chose one mitochondrial gene and one nuclear gene to kind of get a better picture since these wasp species are so closely related. We wanted to make sure we had enough genetic information from two different locus sites to try to get the best picture of like evolutionary change that we could. So we used the mitochondrial one because it has a faster evolutionary rate and is seemingly more used in literature, but it does have some problems with some introgression and since it's only inherited linear or maternally. And then we also used a nuclear marker because they will go over the crossing over, and it should have a better sight of the whole lineage, but it might evolve too slow to actually show change, and since these species are so close. And so that gave us these maximum likelihood trees that you can see here, where we colored them by the region of California that we collected by. And in the 28S region, we have almost two base pairs were different, but we have this one clade kind of out to the right, and then all the other ones kind of falling here. Interestingly, there does seem kind of with our previous iNaturalist observations, it does seem that Eldoridensis seems to be specialized in the California Central Valley, with all of them kind of differentiated, and then the Californian, or I mean the Oregon species and the kind of Oregon California border species seem to be falling to their own thing, which we can kind of see in the mitochondrial where there's a lot more differences. We then decided to not look at it geography wise and then try to do it by host species. And so here we have the Oregon white oak kind of being a way more closely related with the species choosing that. And then we have the California valley oak with the wasp species choosing that. And then here we have our prehensile clade, which goes on Quercus barbifolia, which is the California brambling oak. Oh, sorry. Um, so I did this analysis at the end of my capstone, and we kind of looked and we were looking at it, and seemingly there wasn't enough genetic difference to differentiate them as species. But they do seem to be host specific, and we were able to confirm that phenotypes seemingly are more changing due to temperature rather than just purely gene expression, even though most of the species do seem to have slight differences, especially with prehensile being a standout. Um, we then, for my thesis, wanted to take another approach. Instead of using maximum likelihood trees, we decided to do a Bayesian phylogenetic analysis using BEAST. Um, so this allows us to use several different loci and kind of concatenate them into one single species tree. So we took, and then talking to other researchers, it seems that like cytochrome B and ITS2 is most used for this Bayesian analysis. So we decided to use 4 genes since we already had cytochrome oxidase 1 and 28S. And then using this Bayesian principle, we used the wasp's host tree and then pre-assigned them what species we think they are. So if it was on a Quercus lobata, we would assign it Eldoridensis, and if it was Mollifica, we would assign it uh, Gariana. And then instead of using maximum likelihood, it uses a posterior probability, which basically tells you if two species are had such a common ancestor. Oh, sorry. If two species had a common ancestor not too long ago, they're probably not different species. And if it has a value of over 0.95, that probably means they are the same with a common ancestor, not far enough to distinguish them. And then in doing this analysis, you were also able to get gene trees before the concatenation into a species tree. And we can see very similar things with our two trees up here, with 28S being basically no differences, CO1 having a little bit more variation. And then ITS, which is a nuclear marker as well, has variation in cytochrome B. So we kind of see this kind of like messy trees with all our species kind of everywhere, but eventually we can concatenate them through a bunch of different simulations into each individual species clade. And then using posterior probability, we can kind of differentiate them at that point of the analysis. So after doing all of that, we get eventually this species tree where we have a posterior probability that Mulfica and Prehensa are probably not different species, because their probability of them having the same common ancestor so soon is like almost 1. But Eldoridensis is able to be separated by that sister species group because its posterior probability is actually quite low. Um, but this also wasn't the greatest results because we were only able to get one prehensile species to amplify with all 4 genes. So we decided to take GenBank samples and add that to another Bayesian analysis. And using our GenBank samples, mostly by Nichols et al., who helped collaborate a little on this project, it seems that Malefica and Prehensile are actually separate, just very closely related sister species. And then when we added more Eldoradensis samples, they also seem to be more closely related than the ones we collected, but there is a quite ch high chance that the ones that GenBank are calling Eldoradensis might not be just because they look so similar. But they do seem to be very separated on host tree rather than just the actual Gall morphology. Um, so to kind of like as the end point of our project and what we hope to get out of it is to kind of fix this like taxonomic kind of misunderstanding and also like reporting of these species, where Malefica is only really found on Gariana, Eldoradensis is really only found on Quercus lobata, and Prehensa is really only found on Quercus brevifolia. So overall, our findings are that these are three distinct species. There's strong host specification and some evidence of hybridization in gene flow, which might also be confounded by that oaks hybridize quite quickly. So it's hard to see if a hybrid oak is actually being specialized on or not. And the species boundary, since they are so closely related with their basal clad being 0.99, is understood as porous and not strict, where they probably are interbreeding, but since they're specializing on trees, they are not. Anyways, I'd like to acknowledge my collaborators and mentors that helped me through this project, and also the California Institute of Biodiversity for funding some of the research and also the collection trip to collect all these galls. Any questions?
We have one from Guerin Brown.
Oh, yeah, do I look at the chat or do I?
Yeah, he just unmuted me. Yeah,
Okay.
great talk. It's super fun to hear people talking about oak galls. I had a question because I know that Sinapini is usually cyclically parthenogenic. Um,
Mhm.
are the California species you were looking at, do they have sexual and asexual generations? And is there any noticeable morphological difference between those that you considered?
So, um, there has been no studies on Disholcaspis themselves, but a closely related species, um, in like true cynipids and mirabilis, they do have a asexual stage and a sexual stage, and the sexual stage is the actual stage where they create the gall. Since these species are so small and tiny, it's actually quite hard to like collect them in the field and see if there's actual wasps differences. So for our study and what we were able to do, we were only able to collect gall like wasps out of the galls themselves. So we were only able to see the sexual stage species. But talking to Nichols and talking to my professors, they're fairly certain that there is an asexual step in Distelcaspis as well.
Yeah, okay. That's pretty typical, I guess, for especially for Disholcaspis.
All
But
right, we
yeah,
have to move on
wonderful
to
talk.
Yep,
Thank
the
you.
we
next
need to
speaker.
move
Thank you.
on.
Yeah. Thank you
Thank
so
you,
much.
Carson.
All right. Can you hear me?
Yes, thank you.
Hello. Okay, good. All right. So my name is Linford Otoo. I'm a third year PhD student at Wake Forest University, and my talk is on genetic signatures of emergent DS1 human rotavirus reassortment with WAP8 VP4 genes. So I'll give you a short background. Okay, so rotavirus is an important human pathogen, both in humans and animals, and it was discovered in 1973 by Dr. Ruth Bishop. There are several groups of rotavirus, but I'm particularly interested in rotavirus A because it causes a majority of the disease in humans and animals. So rotavirus is known to cause more than 160,000 deaths per year, especially in developing countries. And rotaviruses do not just affect humans, but also affect a wide range of other organisms as well, other animals as well. The good news is that there are live attenuated vaccines for rotavirus but these vaccines are known to underperform in developing countries, and these vaccines may not protect against some zoonotic strains. Therefore, it's very important to research on the rotavirus epidemiology, evolution, and diversity to inform the next generation of vaccine design. Now, rotavirus, so we'll talk about the structure of the rotavirus. So this is a picture of the rotavirus, which is a triple layer virion particle. It's made up of the outer capsid protein, which is VP4, as a spike protein, then VP7, the yellow VP7, which is also the capsid protein, then the intermediate layer, which is VP6, then the VP2, which is the inner capsid that houses the VP1, which is the polymerase VP1, and VP3 as the capping enzyme, and also houses the 11 segmented double-stranded RNA right here, right in there. Now, this is a gel showing the 11 segmented double-stranded RNA by gel electrophoresis, and the segments are in two forms, which is VPs and NSPs. VP means virion protein, and NSP means non-structural protein. The VP is viral protein because it forms part of the final structure of the virus, but the NSP doesn't form part of the final structure of the virus. It's mainly produced within the cell to enable for to facilitate replication of the virus. And these are their various functions. Now, one mechanism about segmented viruses is the ability to undergo reassortment due to their segmented genome. What I mean is that when there are two different strains, strain A and strain B, and they co-infect the same cell, they are able to shuffle their genomes and end up coming up with this novel strain called the reassortant strain, just like flu. And this reassortant strain has three possible fitness, either an increased fitness or no change or decreased fitness as compared to the parent strain. Now, one part of the rotavirus story that is very important to my work is the current classification system, which takes into consideration the entire 11 segmented, the 11 segments of the genome. So what they did was to assign a letter to each of the segments, and the letter is based on the function of the segment or the protein. So for example, with VP1 is assigned R due to its function as an RNA polymerase all the way to NSP5, NSP6. Then using a percentage nucleotide identity cutoff value, they were able to assign a percentage to how many genotypes can be found within the same genome or the same segment. So for example, with VP1, there are 28 different genotypes of VP1 alone, using a cutoff value of 83%. So they did that for all the other segments as well, and we could found so many different genotypes or more of like genetic diversity of the same segment. So the next thing they did was to bring these letters together to form this system called a genotype constellation. So each of the letters you see here are the segments, and the X you see here is the number of genotype of that particular segment. So the next thing they did was to sequence a lot of the viruses across the globe, and they got to find out that the genotype constellation of humans is in two flavors, which is WA genogroup and DS1 genogroup. So you see that red or green using this constellation, even though there could be many of them, but they were usually only these two. And they also found very rare mixture of the red and the green, and it was called the intergenal group reassortants, where we could see a mixture of both red and green. And they were known to be very rare because out of like 1,000, just find one or two present, and they were not known to spread. So it was assumed that it was hypothesized that these intergenal group reassortants have decreased fitness because they were not found more in the population. So this reassortant viruses have decreased fitness. Now, in 2014, they found 50 intergenous group reassortants out of 107, which shows that it was beginning to emerge more and spread. And they had this particular constellation. It had this VP4 and VP7 and VP4 was from the regular WA strain, which is the all red, sorry, all green. So you could see that as green, and the backbone, which is from VP6 all the way to NSP5, was from the DS1, which is all red. So there was a mixture of both red and green. And we also found more of them in Australia as well. And they also had the strains that we found also had the same constellation as well, which is the VP7 and the VP4. But this time the VP7 changed with a different genotype. But the VP4 right here, the VP4 and the backbone was the same as the previous. And more of these intergenous group reassortants were found across the globe, different places, and they had a very similar pattern of this P8 from the WA, and the backbone was from from DS1. So you could see a mixture of both with the VP7 always changing. So this is what my work is particularly focused on, which is the P8 on a DS1 backbone. So we could see this all over the globe, across every continent, across every country in the world. So could it mean that this intergenous group reassortant, which I would like to call a P8DS1 reassortant, now fit? So this is the overall objective of my work, is to investigate the influence of intermolecular compatibility of rotavirus A, gene reassortment, evolution and emergence. And particularly I'm looking at to identify unique genetic signatures. I'm looking at both nucleotide and amino acid differences of these emergent reassortant strains. Yeah, so I'm looking at the genetic relationship between the two across different locations and across different years. And I'm looking at the genetic signatures between the two. Now, first of all, I compare the VP4 to the typical VP4 to the reassortant VP4, how similar or different are they? And I use phylogenetic tree, and this is I use neighbor joining method here to come up with this result. This for only VP4, and we could clearly see the white dot here is the VP4 of the reassortant, and the black dot you see here is the VP4 for the typical strain. And we could see that majority of the reassortant strain clustered together with very strong bootstrap values, and the non-reassortants also clustered separately, and we labeled it B allele and A allele. And we zoomed in to actually look at that which strains are involved right there. And we could observe that the VP4, regardless of the number of years and location and different VP7, the VP7 that changes, they all clustered together. And however, there were few non-typical VP4, non-typical P8s that clustered together with the reassortant VP4. So we also did the same thing for the other backbones as well, such as from VP6 all the way to NSP5. So we looked at the other internal genes, for example, VP1, even though they look the same, R2 is the same as R2, but is it really the same or is genetically different? So we ran the phylogenetic tree using neighbor joining method, and we found the orange pink one was known to be the G8 from horses. Sorry, from cows, sorry, bovine cows. Yeah, and we also decided to look more into the B allele, and we found the same pattern, that regardless of the years and locations, and they all clustered together right here. And there were also some few non-reassortant R2 or VP1 that clustered together with the reassortant strains. So we decided to look at the full constellation instead of per segment, and we find out that with the typical strains, we found out that most of the segments or its constellation was major red, and the reassortants were known to have a lot of yellow or B type allele. So the non-reassortant had type A alleles, and the reassortant had type B alleles. And we quantified it, we could clearly see that all of them, VP4 all the way to NSP4, had the reassortant had the B allele specifically. So we decided to look at the genetic signatures that differentiates between the B alleles and the A alleles. And we found 6 nucleotide changes that separates the P8 A allele from the P8 B allele. And we found only 2 amino acid changes, which is from lysine to threonine and isoleucine to phenylalanine on the VP4. So the VP4 is divided into VP5 and VP6. So and these 2 amino acids were shown to represent in the A allele as well, but they were actually fixed in the B allele. And these 2 amino acids were found on this domains of the VP4, and which are really known for cell penetration and interaction with the cell. So it could mean possibly that the reassortant strain kind of introduced this mutation in order to better infect the cell, and that led to its spread across the globe. So in summary, since 2012, there has been an emergence of this particular reassortant strains, and it shows that these P8 reassortant are similar to each other, but are different from the non-reassortant strains. And it showed that this type B allele was also found in very low frequency in the non-reassortant allele, and they were high frequency in those particular reassortant strain. There were some nucleotide and amino acid that distinguishes them. And it could show that these reassortant strains originated from an exchange between the pure genogroup strains that combine the type B alleles, which are the yellow alleles. And that is how these segments came together to be able to have higher fitness to spread across the globe. Thank you. And
Thank you
questions?
very much. I do not believe we will have time for questions, but thank you very much for your talk. Ben?
Yeah. Hello there. Okay, so let me share my screen. And it's here. Great. Can everyone see that?
Yep. Thank you.
Great. Okay, well, thanks for inviting me here to this the 2026 evolution of this year. I'm really glad I've been here. And well, let's start this. So it's gene family size expansion and longevity among mammals or in mammals. So, whoops, okay, yes. So to start, what we are checking in this paper that I'm presenting was published last year, 2025. It's pretty much the genomic signatures of longevity. How longevity among mammals it's actually evolving. And in our focal stats in this moment, it's gene family size. Gene families, in essence, like in a very simplistic way, it's pretty much a set of genes that are very similar to each other, and they are put in groups, so we can actually compare them, and they have similar functions and such. And then for these, we were using maximum lifespan potential, which is a very popular proxy for longevity, what does it measure? Pretty much is the longest recorded member of a species or group of members of a species. And this data comes usually from zoos, because in control situations is where they can actually measure the longest lifespan of a species, right? Otherwise, the issue will be that you have other factors intervening into the mixture, which is good for some type of studies, but for the one that we have, probably it's better just to have like in this control situation, so we can actually focus on what is actually influencing the genes mostly, rather than that it was, I don't know, eaten by a predator or something like that. So as you can see, there is a quite large diversity between mammals. We have like, for example, some mice that can live like around 1 year up to 200 years in some other mammals. And well, where we're going to check the genetic aspects there. So we analyzed 46 species with high quality annotations. And then we have these contender traits that we wanted to measure that are traditionally associated with longevity, which is body mass, relative brain size, gestation time, and sexual maturity. And well, we compared them, and interestingly, we found that they correlate with MLSP. Nonetheless, the only one that at least in our sample that correlated with gene family size was relative brain size, which was a bit shocking. But well, that was the result in this case. After the ontology annotations, the gene family annotations that enter into the analysis were around 4,000, and then we conducted a PGLS regression with 2 variables to assess this gene family size dynamic. And what we found, interestingly, we found the gene family size analysis between MLSP corrected by brain size, or relative brain size, and relative brain size corrected by MLSP, both together to correlate that, they share 60% of gene families, which is quite interesting, right? Because that means that the longevity and large brains are actually following a similar path, similar story, an evolutionary path. And well, after that, actually, we wanted to conduct a Go enrichment as part of to know what these genes does, and also to understand maybe if predefined selection maybe would be not involved in this, because maybe we were just seeing random expansions, right? But in this case, no. So what we found, pretty much everything is linked with immune system. And here, actually, this is this list. In this heat map, you can see the different functions. But in this other, it's a bit more like diluted, or well, not diluted, it's like more digested, like in more like functioning. Like we have like genes that are linked to inflammation, love of proteostasis, also like lipid homeostasis, immunosenses, etc., right? So what we found here is that apparently brains are composed by, for example, by postmitotic cells, which do not divide. Well, we need something that takes care of that brain, right? So it's like a shield. We need like a redundant system of maintenance in order to actually keep this brain functioning, and at the same time have enough time to live with it. So longevity, right? And yes. So another interesting thing that we found is that after comparing our database or results, the gene families, and then the genes linked to those gene families associated, we compared it against databases that are already published. And some of them, they have like manually curated genes and such. So they're supposed to be very well reliable in this manner. And interestingly, we found that some genes linked to DNA repair, inflammation, and human longevity associated, etc., they were upregulated. Nonetheless, one aspect that we didn't really love is that autophagy, which is pretty much a very well-known trait or function that it's linked to longevity, it was actually downregulated. and we're not really sure why. But then, well, autophagy, just for in case that someone doesn't know what it is in general, it's pretty much like a recycling system of the cells. So it's like they eat they eat like they eat themselves in some kind of way as a self-maintaining process. And then what else? Then we checked if actually alternative splicing was actually present in our sample. Alternative splicing is like the Swiss knife. It's like a Swiss knife of like for proteins. So we have like one gene that can actually divide itself in different parts, and then it decoding in different proteins with different functions, right? So apparently, in our case, it was actually upregulated compared to random, by chance. And well, so in some interesting parts, so what we're looking at here, it's just one path of the evolution of longevity linked among mammals in these 46 species that we were actually looking at. And what we found is that we have relative brain size, we have maximum lifespan that which they correlate with each other, and they correlate with duplication, like gene family expansion, and also with alternative splicing. It gives us like an immune system, it gives us immune system expansion. And this immune system expansion is actually linked to cellular maintenance and DNA repair, and also other functions that actually help us to live longer, right? So in this case, this is a correlative study, right? So we have to be sometimes a bit careful that it's not a causality. It's not a causality case, right? So, but still, it's like a hint of what is happening. So in general, what we can say is that we have three aspects that we can take home with us at this moment, which is that brain and lifespan are coupled together. So immunity, it's like the housekeeping of the house in this case, and then it helps us to live longer. And that mammals, they share a common story for longevity mechanisms, and in between the gene family expansion, which is something fascinating because that actually allows us to go more in depth and try to find not just focusing in humans, but also how mammals have an impact in their longevity, and maybe even we can actually try to make them to live longer with us, right? And well, I want to say thanks to pretty much my team between University of Bath and the Institute of Ecology in UNAM University, plus also saying thanks to our sponsors like SSCT in Mexico, and also some of the images, or most of the figures used in this presentation were actually drawn by an artist that she's also a biologist called Milkojo. So in case that you want to also collaborate with her, just drop her a message or something like that. And that's pretty much it. If you have any questions, I would be very happy to answer them.
Thank you very much. We will have about 4 minutes for questions. Please post them in the Q&A. I have one of my own, actually.
Go ahead.
Is it correct, or can we reasonably surmise that the ancestral condition for the mammalia as a whole was to have a short lifespan, as in just a few years or less?
Okay, that we can assure that. Well, we could say based on that pretty much everything started with more organocodone in some kind of way. So yeah, I mean, supposedly they were like very short-lived mammals, and then by the pass of the time, they started to gain genes and they started to become bigger and bigger and such, and also their ecology changed. So that started to make us to live longer and also to have different, how can I say it, different ecologies and different behaviors. And behavior probably it's one of the links to actually make brains bigger, like the social brain hypothesis, right? So, and then therefore, in some kind of way, by having bigger brains, also resources have to be given to the brain, and also it's like a fall, right, of different aspects that actually started to make us to live longer, because also we need more time to develop, and etc., etc., etc. It's an interesting story indeed.
Okay, thank you. We do have time for one more question, which I'll read out from the question and answer. How
Sure.
were the species in the phylogeny selected? Was it what was available, or did you try to maximize variation in potential lifespan?
Actually, it was because it's based on what was available at the time, mostly. And also based on genome completeness and those kind of stuff.
Quick follow-up from me, since we have, I think, one more minute.
Mhm.
Um, are there any clades within the mammalia that you would like to sample more of in the future?
Well, of course, for example, bats are a great example because they are an amazing group to actually study longevity, as they have a very small size, like a body size, and also very and long lifespan, right? So that would be a good option.
Okay. We are almost ready, I think, for Sienna to share.
Yes. Thank you very much.
Thank you very much. All right. Just give it a little bit longer.
Yes, let me pull up my.
Feel free to go ahead and start sharing.
Yeah, I'm trying to. Sorry, just trying to share this specific window and it's not popping up. Okay. Okay, one second.
All right.
Okay.
Let's go ahead and get started.
Does that look okay?
I think so.
Yep.
Yeah.
Okay. Great. Okay. All right. Thank you so much for having me, everybody. I'm Siena McKim. I'm a fifth year PhD candidate at UC Santa Barbara, and I'm excited to talk to you today about my research. So I just want to start off with some background. Some of you might not know exactly what silk is. So silk is more than just something that's like secreted out of the body, kind of like a mucus. Silk actually requires a really radical transformation. So in the body, if we can imagine this body part as a leg, it starts off as a liquid feedstock, and it's made out of these long repetitive proteins that start as a liquid, and then as it comes out of the body, if you look at the tip of the leg, it transforms into a solid thread. So you might be familiar with like spider webs, like those threads are these like cylindrical threads that are made out of these crystalline domains that give it strength. The important thing is that the silk is going from a liquid to a solid, so this requires really specific morphology to make this transformation happen. And silk is really common in arthropods. It's used in many different groups for daily functions like reproduction, for feeding, for protection. So it's a really critical adaptation that's evolved in arthropods. In fact, spun silk has evolved
Sienna,
over 30 times.
are
Yeah,
are you advancing your slides?
yes.
Okay, we're not
Are you
seeing
not?
that.
Oh gosh. Okay. Really? Okay. Sorry, I don't know why that would be. If I have it like this, can you see it?
Yes.
Okay, we're just gonna leave it like that then. Sorry about that. Just to recap, silk starts as liquid in the body, and then you can see here it comes out as a thread. It's in all these arthropod groups. And spun silk has evolved 30 times in arthropods. So you might be familiar with arachnids. It's evolved in spiders and mites and others, in crustaceans, in marine and terrestrial ones, and then 23 times in insects. So it's a really widespread adaptation that's evolved in these groups. If we focus specifically on crustaceans, it's evolved 6 times. And in my research, I'm specifically studying amphipods, and it's evolved twice in that group. If you're not familiar with amphipods, there's these really small marine crustaceans that are found around the world, and they're generally very small. They're only about a couple millimeters long, but they will create these really diverse structures with their silk, like these rods and tubes that they use for feeding and for protection and reproduction. And originally, there was a hypothesis that silk evolved twice in amphipods. So first, so in the amphipods on the top here, and then the coryphioids on the bottom. However, the silk spinning morphology, so what they're using to actually make the silk, hasn't really been well documented. So in the amphipods, there's almost been no studies on what the silk spinning systems look like. And in the coryphioids, it's a really big, diverse group. And so that the diversity of the silk spinning systems hasn't really been documented in that group. And in general, the phylogenetic relationships of amphipods in general, but specifically these groups, is not really well resolved. So my research has two driving questions. Do the coryphids and the amphipods have a similar silk spinning system? And then are there any characteristics of amphipod leg morphology that might actually pre-adapt these lineages to evolving silk spinning systems? So is there something that was present in the ancestor that might have allowed for the origin of silk spinning specific morphology? So to do this, I compared the silk spinning systems between these two groups, the amphipods and the coryphioids. And what I did was I took off the silk spinning legs, so the third and fourth pair of legs here. And then I first was looking at the glandular system, so in the upper tissue of the leg. And then zooming in on the tips of the leg, this is actually where the silk is being transformed. So this is the silk extrusion morphology. And this is actually telling us how the silk is being transformed from a liquid to solid. So it's a really important structure to keep in mind. And so not only comparing these two groups, I'm also interested in what the evolution of these traits are. And so I also sampled outgroups from these groups to see if there's any glandular systems in those legs. So to do this, I did a lot of morphology work. I stained all these legs with fluorescent stains, non-fluorescent stains, and did various imaging methods such as confocal microscopy, light microscopy, scanning electron microscopy, and even some micro CT scanning. In total, I looked at over 55 species, representing the diversity that's in those groups, and then I looked at over 400 legs. So with the amphipods, to get a more phylogenetic perspective of what's happening, I did some phylogenetic estimation using maximum likelihood and also Bayesian inference. The phylogeny you're seeing here is the Bayesian inference tree. And you can see here that amphipods are a monophyletic group, which is great for this analysis. And then I used the closest outgroups, the cynopids, which are like these helmet head amphipods. In the coryphids, I actually worked really closely with Brittany Cummings, my collaborator, who did most of the phylogenetic estimation methods. But you can see here, the coryphioids are also a monophyletic group. And then we did some sampling of non-silk spinning amphipods. So now that I have all the morphology traits and also the phylogenetic framework, I put those together. So for the morphology, what I found was that in both the amphipods and the crofioids, they use two gland system, which means they have two gland types that are working together throughout the leg tissue, and they travel down the leg and secrete. So you can find the first gland type and the second gland type here. They're really interesting looking, and I wish I had more time to talk about them. But the takeaway is that both of the lineages had these two gland types, which is kind of unique when you look at other silk spinning arthropods. There's differences in the distribution of these glands across the legs. In the amphipods, you could actually find the glands in all of the legs, not just the third and fourth legs, which is really unexpected and fascinating. When we look at the very tip of the leg, where that silk is actually being transformed, there's this really important structure called the chamber, and this is where the secretions are traveling to, and then they likely transform the silk dramatically. And this is only found in amphipods. This structure is not found in any other silk spinning arthropod group as far as I'm aware. And if we look at the external morphology, you can see there's kind of similarities. Amphipods have multiple pores at the tip of their leg here, and then the crofioids have one main pore at the tip, and they both have like these extra sites where secretions come out of. But in general, both of these lineages had this reservoir chamber that seems to be really important for silk transformation, and have multiple modes of extruding these silk products. So there are differences in how these silks are actually interacting once they're outside of the body. And so there's a lot more work that needs to be done in exploring that. So then in our non-silk spinning outgroup, I found that actually that second gland type was found in these groups, and it was found in every single species that I examined, which is really unexpected. So they have these second gland types in the same legs, but the difference is that they don't have the silk extrusion morphology at the tip of their leg. It seems like they are secreting whatever these glands are making out of hairs or these seedae that are on the legs. So it potentially suggests that they're making maybe a similar secretion as these silk spinning amphipods, but they're not having the morphology to actually transform it. So if we look at the ancestral state reconstruction analysis for these traits, so just looking at gland type 1 and 2, we can see in the amphipods that in the common ancestor of Amphipolicidae, that this first gland type likely originated in that common ancestor, and that this second gland type exists in the outgroups. And then in crofioids, it was a little interesting. It seems like from this ancestral state reconstruction analysis that not only the crofioid common ancestor might have had these two gland types, but actually the ancestor that predates that. This is something really interesting to explore further. But as you can see here, these second gland types are found in all of these groups. And so from all of this work, what we can actually gather is this updated hypothesis. So originally I said that these two groups were convergently evolving these silk spinning glands, and it's still kind of the case, but it seems like that there is this ancestral trait of having these type 2 glands in the legs that might be secreting a similar product, but they didn't have the extrusion morphology to actually transform the secretions. And then within the lineages of the common ancestor of Amphipolicidae and the crofioids, that this morphology for actually spinning silk and this first gland type you can see in orange actually originated. So there's there is this similarity in this origin potentially of these morphologies that allow for the spinning of silk. And so this work is really helpful for giving us an understanding of the evolution of silk spinning systems in arthropods. As I said earlier, it's evolved so many times, but actually in many of those groups, we don't really fully understand like what the silk spinning systems are and how they're actually transforming silk. So this is a new perspective on those silk spinning systems, especially because in these marine crustaceans, those systems are really overlooked. So it's bringing new light into how those silk spinning systems might actually have evolved in arthropods. And with that, I just want to thank everybody for listening. Thank you for organizing and for all your help, for my collaborators and everybody that's gotten me through this project, for all of my funding sources. And if this work interests you at all, you can scan the QR code to link to my ORCID ID and with all my papers. And if you're further interested, you can email me and I'll take questions now. Thank you so much.
Thank you very much. Do we have any questions to be posted in the Q&A? I have one. What was the analytical framework that you used for the ancestral state reconstruction?
So the are you talking about like the models that I used or like what specific part?
Just the program that you used for that part
Yes.
of the analyses.
Yeah, I used REdisk within Core HMM.
Okay.
Yes, yeah. Yeah. I think there is another question.
I have a quick question. I think we have a minute if that's okay.
Yeah.
I was wondering if there's like a biological advantage to having this like second gland, because they seem to like that one has it everywhere, even though it's not secreting there, and then there are all of these with them. If you know.
Yeah, so our original hypothesis was that these glands are interacting with each other. So potentially in the crepioids, which are a little more well studied, some people have kind of speculated that these two gland products are actually mixing together almost like an epoxy. And then when it comes out of the body, it's producing a new kind of like product that's been altered. So that's kind of one speculation. In our research, we actually found that the glands and the ducts systems are separate. They don't seem to actually be mixing. I didn't show, but there's a duct that bypasses the reservoir chamber, and I think that's it's producing like a different kind of secretion that's not undergoing the same transformation in the chamber. So yeah, it's really interesting why, yeah, those two glands are definitely making different products, but how they're interacting is still definitely a point of interest for my research.
Thanks again, Sienna. And we can now switch to Kevin. Kevin, please go ahead and share your slides, and we can start in a few seconds.
Sure.
All right, please go ahead and begin.
All right, thank you very much. I'm Kevin Liu. I'm a faculty member in the Department of Computer Science and Engineering and also the Ecology, Evolution, and Behavior program at Michigan State. Here to give a talk on behalf of my co-author and I, Meijun Gall, who's, quick ad, basically going to graduate very soon. So if you're on the lookout for a really excellent researcher in computational phylogenetics, who's got lots of different interests, including in AI, applications of AI to phylogenetics, please do reach out to me. So basically, I'm going to talk about a work which was published just last year, where we're essentially bringing together a bunch of different computational ideas to address kind of an interesting problem, trying to detect and map local model violations during phylogenetic analysis and other forms of biomolecular sequence analysis. And so we're using a bunch of kind of interesting ideas that haven't been applied to this task so far. And I'm going to talk a little bit about exactly the problem and kind of how it can arise. It's actually been hinted at during this session. So this has been a kind of a nice introduction, all the different ways in which computational phylogenetics are used, but then a lot of the issues that can crop up. And then I'll talk about our solution to this problem and kind of how we study this solution. And I'll wrap up with just some thoughts. Okay, so a lot of the talks today have covered how statistical approaches are really quite popular, quite widely used to perform phylogenetic analysis and other related forms of evolutionary analyses of biomolecular sequence data. And so these statistical methods, I think pretty much everything today, and mostly the popular ones throughout, they're reliant on explicit parametric models of evolution. And so when you're applying such model-based approaches, an essential prerequisite, an assumption is that the model that you're working with is applicable to the data that you're studying the evolutionary processes that you're thinking about. And so when that's not really quite true, and depending on kind of the degree to which that assumption is violated, some issues can crop up, in particular model violation. And so there's really kind of two basic types that have been heavily studied in the literature. I think the one that's been typically more addressed is a so-called global model violation, where essentially one model is assumed to apply to all the different parts of your data. So for example, the different sites in the alignment that you're trying to perform phylogenetic analysis on. And if that model doesn't fit quite right for all the different sites or just parts of your data that are under study, that would be a global model violation. In contrast, if you've got essentially parts of your data where a model violation is present, this can be considered to be a local model violation. And so you can have, in fact, multiple such local model violations due to a variety of different factors when the model of evolution that you're working with for your data isn't quite applicable to a region or different regions within your data. And that's what I'm going to focus on right now. And so, in fact, there's a lot of different reasons why these local model violations can occur, some of which have been discussed, during today's talk. So, for example, you can have non-tree-like evolutionary processes, which were briefly hinted at during the first talk. If you've got things like hybridization, introgression, horizontal gene transfer, and so on, and these types of other reticulate evolutionary processes, this can cause kind of local model violations. In addition, you can have natural selection in different forms, genetic recombination, and also combinations of these and other processes can also violate your typically used models for your global data analysis. And so there's a bunch of different ways in which you can deal with this. And I would say probably the typical traditional ways are probably one of these two following approaches. The first would just be kind of the traditional scientific method. You have an initial model, you perform your analysis, you assess the extent to which it kind of makes sense or doesn't, and then you kind of have an iterative model refinement approach. And this is kind of a bit more traditional. And then there are kind of more automated computational approaches that, under the assumption of a particular model that's applicable to your data, they'll try to go and find model violations. And so there's a bunch of different work along these lines. But with respect to both of these approaches, they are kind of a few different kind of factors that, kind of to some extent impact applicability and throughput. One is the need for assuming that a specific model might be present to cause the local model violations. The second is just typically the approaches for the automated algorithms that have been developed tend to be developed for a specific task. And if you're using machine learning, there's typically other assumptions baked into that, such as the need for labeled training data. And so our project really begins by trying to think if we can move beyond these limitations, these features. And so we want an approach that can both detect and map local model violations without requiring specific assumptions about local models that or just specific sources of model heterogeneity. And that can be applicable to a variety of different tasks, not just a specific closely related set. And so this is kind of our formulation of this problem. The input being a multiple sequence alignment. You have n sequences, typically representing different taxa. You have case sites in your alignment. And then you basically have the same requirements as a traditional phylogenetic analysis. Let's say that you have a global model that you think applies to all of the different parts of your data. Then you also have a model-based method. And then what you want to do is basically you want to classify all the different sites and regions within your MSA as to whether or not they are basically evolving under one or a different model. And so this classification problem, we basically require that the user specify that a specific number of assumed models might be present. So for example, here with Z, this being the parameter, if you set Z is equal to 2, then you assume that one model, maybe the background model, applies to most of your data, and then you might have one specific model which is kind of locally applicable to just specific parts and causes one local model violations. For the results that I'll talk about today, just briefly, I'll be focusing mainly on the setting where Z is equal to 2. I'm looking for basically a background model and maybe one specific local model, although the study, we actually explore kind of more models and sources of local model violations beyond 2. Here's just an illustrated overview of our computational approach. And kind of only a few things are really kind of interesting and worthwhile to point out. The first of which is this idea that if we use resampling, statistical resampling techniques, so if you're familiar with something like bootstrap resampling as it's used for phylogenetic bootstrap, this is one form of statistical resampling. If you apply that locally within your data to get a local pseudo replicate of your data, and then essentially repeat your analysis just on that local part. And the key idea is if you compare that local reestimate on that local pseudo replicate versus the original estimate across your entire data set, the extent to which that differs is a key clue as to whether or not there is a local model violation. That's pretty much it. That in a nutshell is our method, which we refer to as a method for resampling and visual evaluation or reveal for trying to detect and map local model violations. The specific way in which we do that, there's a few kind of interesting twists that we add on top of that. We essentially want to represent the local reestimates and how much they disagree with each other and the global estimate in the form of essentially a summary, being an image-based summary, to which we can then apply techniques from computer vision to try to summarize regions of disagreement from the fine scale disagreement patterns. That's kind of another interesting twist that we added on top of this. That's basically it. I'm going to super quickly kind of run through a little bit of the technical details about how we do this, although I basically boiled down kind of the interesting technical contributions already in the last minute. So there's essentially three stages. In the first stage, we need to do the local resampling, statistical resampling that I just said to get local pseudo replicates. We tried to use a new method that we developed a few years back, something called a random walk resampling method to do this local resampling while trying to retain kind of sequential ordering of our sequence data. So then after that point, you basically use your model-based method for the global analysis to re-estimate on the local pseudo-replicate to get these local re-estimates. And then you compare them to each other as well as the global estimate. And we look at a few different statistics to assess disagreement of these local re-estimates. They look at things like topological disagreement as well as branch length distributions. And then we take all of these raw statistics and we essentially summarize them into one single representation, basically an image. We just represent them essentially in a heat map in an image. And that image then allows us to apply unsupervised kind of clustering techniques to basically try to summarize the fine scale site-based raw disagreement statistics into regional patterns of local model violations. And so we use a really super simplistic traditional classical clustering technique for this. K means nothing special, where K is set equal to Z. That's basically it. And so we conducted some performance benchmarking experiments to study this reveal framework to see if we could detect and map local model violations due to a variety of different causes. So I'm just going to talk about the simulation study part of the experimental study. We also performed some empirical data set benchmarking as well. If you're interested, you could consult with the publication about that. Briefly, the simulation side of the performance benchmarking looked at the following different causes. We had, let's see, basically gene tree heterogeneity, different causes of that, summarized into these H set of model conditions. We looked at genetic recombination. We looked at non-tree-like evolution due to introgression. We looked at kind of natural selection and selective sweeps. Then we also looked at mixtures of these different causes. And so here is just a single figure showing you just a little bit about the performance outcomes for reveal, trying to detect and map the different local model violations that occurred due to these different causative factors in our simulation experiments. On the top, you basically have a type 1 error assessment in the form of precision. Higher is better along the vertical axis. And on the bottom is recall, trying to assess type 2 error using recall. And then you have these different model condition sets, H, R, I, S, H being gene tree heterogeneity height, R is recombination, I is introgression, S is selection along the columns. And then within each column, we're varying kind of the intensity of the local model violations. Long story short, precision and recall were basically around 85% or better, pretty much consistently, at least across this small set of conditions that we studied. And that's basically all that I wanted to say about this. There's a whole bunch of other kind of experiments that we ran, at least in the simulation study side, looking at kind of ablation experiments and just other factors that could arise. In addition to having kind of the multiple local model violations within a single data set. And basically the performance is roughly consistent with what you see here. Okay. So in summary, what I talked about is essentially a new computational framework that essentially is a proof of concept showing that you can develop a pretty general purpose, flexible framework for detecting and mapping local model violations due to a variety of different sources without any specific simplifying assumptions about what specific causes might be giving rise to those local model violations. You really don't need any modeling assumptions beyond that needed for a traditional global analysis. There's other benefits as well. Pretty much our technique has the benefit that it doesn't require labeled data. It's not a supervised statistical learning technique. It's fully unsupervised. And we suspect that actually this approach is probably a lot more generally applicable to a lot of different tasks beyond just, in our case, looking at phylogenetic reconstruction from aligned sequence data. You could probably use it, sorry, unaligned sequence data. You could probably use it for a lot of different phylogenetic inference and learning tasks, and perhaps even beyond that as well. So this is kind of just our first kind of attempt at working on this task. We have a lot of other directions to work on this, including looking at different applications of some of these other ideas, as well as trying to apply more sophisticated techniques. For example, I mentioned the very last stage, trying to basically summarize regional patterns into a final output from the fine scale disagreement statistics. There's probably a lot more sophisticated techniques that could be applied there. Lots of my colleagues in computer vision have thoughts on that, so maybe we'll look into that soon enough. With that, I'd like to thank all the funding agencies and institutions that have supported us in this project. Here's my URL. You can find my contact info there, as well as a GitLab repository with all of the study scripts and data for this project. So thank you very much for your attention.
Thank you very much. I don't believe we will have time for questions. Kate, could you go ahead and get your slides ready, please? All right. Might as well go ahead and get started, I think.
Awesome. Hi everyone, I'm Kate Truman. I'm a PhD candidate at the University of Canterbury in New Zealand, and this is joint work with my supervisors, Alex and Sasha, as well as with Bethany Allen in Germany and Ugna Stoltz at ETH Zurich in Switzerland. So today I'm talking about Skyline intervals and how they impact diversification rate estimates under the stratigraphic ranges fossilized birth-death model. And that's a lot of words and terms, so I'll talk through what that means shortly. So the motivation for this is that we wanted to implement the Skyline stratigraphic ranges fossilized birth-death model in Beast2. So Beast2 is a very popular software for doing phylogenetic analyses. And the Skyline model, so when I say Skyline, I mean piecewise constant rates, and it extends an existing implementation of this model in Beast, which uses constant rates rather than piecewise constant rates. And our goal is to infer evolutionary histories using the Bayesian inference scheme. And we want to infer both diversification rates and phylogenetic trees. And in this case study that I'll be talking about today, we're looking at a data set of crocodilians, so crocodiles and alligators. So when I talk about a stratigraphic range, what do I mean? A stratigraphic range tree allows us to directly model when species start and end. We can include fossil samples, multiple samples from the same species, and we can label that they are in fact from the same species and not related species. So the stratigraphic range model was described by Stadler and co-authors in 2018. And you can see here that on this phylogenetic tree, we have these blue areas here, which are our stratigraphic ranges. But first, I'll just talk a little bit more generally about the parameters in the tree and in our model. So the event types that we can have in this birth-death process are these diversification rates, which are time-dependent Poisson processes. So we have birth, death, and sampling. So firstly, considering birth, we say that one parent branch or lineage can speciate or give birth to a new daughter lineage, while the parent branch also continues on. And this occurs at this rate lambda of 2. So we can see that in this diagram, we have this branch 1, and we have a speciation event where it gives birth to branch 3, and both of these lineages continue on until they either reach the present or go extinct at some other time, which brings us to our second event type, which is extinction. So we say that species can go extinct at this rate mu of t, and we can see that in this phylogenetic tree that lineages 2, 3, and 4 don't reach the present at time 0, but they die out sometime before this. Finally, we can sample lineages at this rate psi of t, so we have these diamonds, which are now coloured red, to denote where we have these samples. And we can see that we can have a number of different samples. For each lineage, we might only have one fossil sample for a lineage, or we can have many. And we want to be able to include that in our results because it gives us information about how long that species was alive for. Note that when we talk about sampling here, we're not considering samples of extant species at the present, but rather just extinct species. So in a macroevolutionary setting, we're talking about our fossil samples. So a stratigraphic range is the full period of time over which we have fossil samples for a given species. So we've highlighted them in blue here. As I said, we may only have one fossil sample for a species, so a stratigraphic range could just be one sample. Or we can see here, for example, for lineage 1, that we have this much longer stratigraphic range between the first and the last fossil sample of that lineage, and we want to include this information in our analysis. So in terms of talking about a Skyline model, we allow our evolutionary rates, so our birth, our death, and our sampling, to take a piecewise constant form. So we say that there are I intervals where our rates can change at times t0, t1, and so on up to ti. So if we look at the same example tree, we have our origin occurring at t0, and we'll have some constant rates, birth, death, and sampling, up until a boundary of t1, and then we have a different set of constant rates. So we have these piecewise constant rates that can vary, that can change at whenever we have a boundary point, and we can arbitrarily define those how we would like. So sort of a natural question is, why are we looking at a Skyline model and allowing the rates to change in this manner? We know that climate change can affect speciation rates, and it's well accepted by paleontologists that diversification rates are non-constant. So even though having constant rates is a really good initial step in a model implementation because it's nice and simple and easy to infer, piecewise constant rates allow us more flexibility in trying to work out what our diversification rates are. Then a following question is, well, piecewise constant is still fairly restrictive. Why aren't we, say, using fully time-dependent rates? And the answer is that we probably don't have enough data to reliably infer that and distinguish between many different possible rate parameters. So we just focus on piecewise constant for now. So in terms of the implementation, there was an existing implementation for BEAST for the constant rates, which is available in the S-Ranges package. This was written by my supervisor, Sasha, and further developed by Ugna Stoltz at ETH Zurich as part of her PhD. And as part of my PhD, I've extended that to allow the Skyline model. And this works by combining the S-Ranges code material with the BD Sky package to allow for these Skyline rates. In terms of the data set that I'll be talking about, we consider a data set of crocodilians. So that is, we have these crocodiles and these alligators. This is a data set where we have, I think, 92 extinct species and 25 extant species. And we can see that the root of the tree is going back to about 150 million years ago. I won't talk too much about the actual trees that we inferred, just for length of time of the talk today. We did also look at this. But the important thing is that the phylogeny is disputed a bit for crocodilians because there are possible homoplasia occurring between various groups. But this is also a really cool data set to look at in terms of our stratigraphic ranges because we often have multiple fossil samples for the same species. So I've said that we can set arbitrary intervals, and we do actually have to choose what those intervals are going to be. So in this case study, we compared the diversification rate estimates achieved under that constant rate model, where we don't allow the rates to change, versus two different sets of Skyline intervals. One of these we refer to as an equal analysis, and in this case we set boundary points at 25 million years, and then every 25 million years back in time, so 25, 50, 75, 100, and 125 million years. And that sets intervals that are of the same length. And then the other option that we chose is what we refer to as chronostratigraphic. And in this case, we set the boundary points based on what is recognized as being chronostratigraphic boundaries of where species and epochs begin and end. So you can see on the right of this slide, we have the figure of the International Chronostratigraphic Chart, and we've got arrows at the points where we introduce these interval boundaries. Note that this creates intervals that are quite different lengths. So for example, our shortest interval is from 2.58 million years ago until the present. So that interval is only about 2.5 million years in length compared to the previous interval at the start of the Miocene up to the start of the Quaternary period, which is about 20 million years in length. So we're now looking at quite different interval lengths, but we've tried to pick the boundaries based on established time periods. So in terms of the actual analysis that we ran in BEAST 2, we use a Markov chain Monte Carlo analysis with a chain length of 600 million for each analysis. And this results in effective sample sizes of at least 150 for each parameter in each analysis. Ideally, this would be a bit higher, and we would also be comparing the distributions to check for proper convergence. This is something that we're running at the moment, so these results that I'm presenting here are just our initial results, and we're confirming them currently. So in terms of what we found, here are the estimates for, so we have on the bottom, on the left we have birth and below it death, and then on the right sampling and diversification. Diversification is calculated as birth minus death. And in these figures we have the constant estimates, so what we'd get without any piecewise constant intervals, as shown in red. The shaded areas are the 95 highest posterior density regions of the estimates, and then the bolded darker lines are the median estimates for each parameter. So we have the constant estimates in red, the green and values are the chronostratigraphic intervals, so those ones where we're doing it based on established time periods, and equal intervals of the 25 million years in length is shown in blue. And what we can see from this diagram is that we're getting much greater uncertainty, for instance, towards the further back in time that we go for our equal estimates, and that the regions for chronostratigraphic intervals and constant rate estimates are much smaller. There is a lot of overlap in the estimates that we get, but there are also periods of time where for certain parameters there is no overlap. So for example, if we look at sampling, and we can see that we have this estimate under the constant model of approximately 0.05 for the constant rate. And if we look between 100 and 125 million years, for example, there's absolutely no overlap between that constant estimate and the chronostratigraphic and equal estimates. We can also see that if we're looking close to the present, that we get quite different results looking at chronostratigraphic intervals compared to the other results. For instance, we have a very high sampling rate compared to the other two models, and our diversification rate that we estimate is very negative. It's about negative 0.4 as the median, compared to rates much closer to 0 under the other two models. So we can see that based on which intervals were chosen, we can make quite different conclusions based on the estimates that we reach. Of course, these are only two possible Skyline intervals that we've chosen, and we've tried to choose interval boundaries such that the number of intervals is roughly even, but there are many different possible values that we could potentially choose. So in conclusion, the Skyline stratigraphic range fossilized birth death model is a valuable model for our phylogenetic analyses because it allows us to include this explicit information that samples are from the same species, and that we can infer a diversification rate estimates for specific intervals and see that they change depending on the interval boundary that we look at. But from analysis to analysis, the estimates that we get can be quite different, and so we need to take care in both choosing those interval boundaries and further look into what makes a good choice of interval boundaries for this. Finally, I want to acknowledge that we are putting this into a manuscript at the moment, so to acknowledge the co-authors of this manuscript, so Bethany Allen, Ugna Stoltz, my supervisors Alex and Sasha, and also Timothy Vaughan and Tanya Stadler. I haven't talked about the philologies that we got for in order to make the presentation short enough, but I'm happy to talk about that if anyone has any questions about it. So thank you.
Thank you very much. We do have time for questions, so please, everyone, feel free to post those in the question and answer. I do have one, and my apologies if I missed this, but is there a particular reason to, so far as you can tell, a particular reason to prefer the chronostratigraphic approach to the equal intervals approach, or vice versa, or is that just not known yet?
I mean, it's not known with complete certainty yet. Partly, if we go back and we look at the tree, so this is inferred phylogeny for the same data set under this paper by Lenny and Yates, so their results are a little bit different. But the uncertainty in the intervals depends quite a lot on how many fossil samples you have. And actually, if I go and I find one of these phylogenies that we have, so say, for example, this is our equal, the phylogeny that we get is a summary tree under the equal analysis. And we've got this boundary, so the white background is an interval and then grey is a different interval and so on. And you can see that the position of just how many samples we have in each interval can be quite different. So, for example, I said that there was a lot of uncertainty back for the oldest intervals for the equal analysis. And if we look at that, we can see that there's hardly any samples in these intervals. So it's partly due to that. Partly, I think equal analyses are easier to think about in terms of it just feeling like we're comparing the same length of time. But that doesn't necessarily make sense in terms of if we know that there should have been, for instance, a period of mass extinction that was in the middle, then that might be impacting our results quite a bit. So I think you kind of want to have sort of a bit of both, that you need to include information that you already know about the boundaries, but the chronostratigraphic intervals that we've used here are probably a bit too uneven.
Okay, thank you very much. And I believe that concludes this session, Phylogenetics 2. Thank you all for presenting, and thanks to everyone for attending.
Thank you very much.
Thank you.
Thank you.
Thank you.
And so I'm not, Kinga and Zachary, I'm not sure which one of you is the main host now, but at this point you can click on the little leave button and close the meeting for everyone. And
Okay.
thanks. Thank you.